cells with similar structure and function and are working together to perform some type of task in the human body. There are 4 basic types of tissue. We are going to start with epithelial tissue. We'll go on to connective, muscle, and nervous tissue. Epithelial tissue has a couple of characteristics that are unique to epithelial tissue. One is that it's composed almost entirely of cells, meaning that it doesn't have a lot of non-living parts to it, and we call that cellularity. It also has special contacts that allow it to form a continuous sheet, and it is considered polar. It has a polarity, which means that it has one exposed surface that is not touching anything, and then a basal surface which is attached to connective tissue, which leads us to the 4th characteristic, which is supported by connective tissue, and it is always going to be attached to connective tissue somewhere in the body via a basement membrane. It is considered to be avascular, meaning that it does not have a good blood supply, and but it is innervated, which is supplied by nerve fibers. So you have receptors that are interpreting what is going on, but it does not get its own blood supply. So that means that it has to get all of its nutrients and oxygen via diffusion. And it's also regenerative. It rapidly replaces all of its lost cells by cell division. When we're classifying epithelia, we're looking at the layers. If it has one single layer of cells, we call it simple. And if it has many or more than one layer of cells, we call it stratified. There are 3 shapes to the cells in epithelial tissue. The first is considered squamous, which is a squished cell, cuboidal, which is cube-shaped, and then columnar, which is column-shaped. The first type of epithelial tissue we'll talk about is simple squamous, meaning that it has one layer, simple, and it's squished. The cell shape is squished. When you're looking at simple types of epithelial tissue, a lot of times things can pass in and out very quickly because it is only one layer. So the function of simple squamous is diffusion and filtration. Filtration is where things are forced across the membrane with pressure. They also provide a slick friction reducing lining, and you'll find them in a lot of different places in your body, in your kidney, lining of the heart, blood vessels, etc. This is a picture of the simple squamous tissue. We are going to be looking at all of these underneath the microscope, and I will kind of explain what you are looking at next time in class. The second type is simple cuboidal. Simple meaning one layer, cuboidal meaning cube-shaped. One of the unique characteristics of simple cuboidal is that the nucleus is right in the middle of the cell. And these help to function in secretion and absorption, and you're going to find them in ducts and glands. Again, this is a picture of simple cuboidal cells, and I will explain what you're looking at next time. Simple columnar is a single layer of column-shaped cells. When you get into the simple columnar, you often will find that they are ciliated, meaning that they have cilia on the apical surface, and cilia moves things. That's its function. And so a lot of times if they have cilia, they're going to have goblet cells, and goblet cells secrete mucus, and then the cilia will move the mucus in one direction across the surface of that tissue. It functions in absorption and secretion. There are some non-ciliated types where there's no mucus involved, and they line the digestive tract and the gallbladder. But the ciliated type, when you have mucus involved in anything, it is to trap things from getting into the body, or try to keep bad things out. And it's also there to protect the lining of whatever. And so the ciliated type you find in your bronchi, which are in your lungs, and then some other places. And this is a picture of the simple columnar epithelial tissue, and you can see that in this case there are some, there's little cilia at the top, and then you can see the cells and the nuclei. And again, we'll be looking at all of this underneath the microscope.